Good morning, everyone. First of all, I would like to thank the scientific committee and the partners for allowing me to present my results. So my topic is dealing with carbamylation of proteins, a molecular aspect of skin aging. So proteins over their life, they are exposed to post-translational modifications. So we can call them young at the beginning and old at the end of their life. And so first, they are subjected to enzymatic modifications, which are reversible and necessary for their structure and their functions. We can, for example, cite methylation, phosphorylation, acetylation. And later, they are exposed to non-enzymatic modifications, so after minutes, hours, even years. And these modifications are irreversible. And they are responsible for the loss of their structure and their function. Among them, we can cite oxidation due to reactive oxygen species, very well known, glycation due to reducing sugar, and also carbamylation. So I'm going to focus on these two modifications. Carbamylation is due to the binding of the isocyanic acid to free amino groups of proteins. And when this binding occurs on the epsilon NH2 of the lysine residues, it generates the carbamylated lysine, also called homocitrullin. So all of these products are called CDPs for carbamylation-derived products. This molecule, isocyanic acid, mainly comes from the spontaneous dissociation of urea in the blood. But it might come from, thiocyanate through the reaction with the enzyme myeloperoxidase or from atmosphere. But these two sources are very minor. Glycation is not a one-step process, but a successive process involving many reactions. So briefly, a reducing sugar will bind on the amino groups of proteins, generating a Schiff base and then an animatory products. And in later steps, these products are exposed to further reactions like oxidations, cleavages, crosslinks, leading to the formation of a wide group of AGEs, advanced glycation end products. So many of them are very well known, but I can cite the CML, the carboxymethyl-lysine, or the pantozidine. These modifications have many consequences. So first, they are responsible for the alteration of the structure of proteins. This is the case, for example, of type 1 collagen. This modification alters the fibers. So you can see here on the top, the native type 1 collagen, and on the bottom, the carbamylated type 1 collagen. So we can clearly see that the network is very disorganized. And consequently, these modifications generate an increase of the stiffness and an increase of the mechanical resistance of the fibers. They are also responsible of inappropriate cell responses. For example, there is an increase of the inflammatory activity of immune cells, an increase of the MMP's secretion, but also a decrease of the senescence of the fibroblasts, the decrease of their proliferation, an increase of their apoptosis, or also a decrease of the matrix synthesis, matrix proteins. So glycation has been widely investigated over the last decades, and especially because 20 years ago, even more, glycation has been described as aging process because the CML and the pathogen increased over time in skin or skin type 1 collagen. But about carbamylation, much less data are existing. So that's why with my team, we suggested to study carbamylation over skin aging. So for this purpose, we extracted skin from 3 different mammalian species with 3 different lifespan. So around 75 years for human, 20 years for bovine, and 2 years for mice. We extracted skin samples and we divided these samples in 3 sub-samples. So the first one is the total extract. The second one was submitted to an extraction and the purification of type 1 collagen. And the third one was subjected to the extraction and the purification of elastin. And from there, we quantified the homocitrullin, so the carbamylated product, and the CML, the glycated product, to compare these 2 modifications by mass spectrometry. And we also evaluate the carbamylation by immunofluorescence. So first, let's see the results from the skin extract. So in plain color, you can see, in plain color, you can see that there is an increase of homocitrullin concentration, so in all studied species, around by 11-fold in mice and bovine, and 10-fold in human. And in squared color, here, you can see an increase also of the CML, but in a less extent, so by 2-fold in mice and bovines, and 6-fold in humans. So we can say that carbamylation is a physiological process, and that the homocitrullin formation is more important than the CML formation. But we cannot say that carbamylation is more important than glycation, because we observe just 2 products. But at least we can say that carbamylation is as important as glycation. Then we pulled this 3 graphs into 1 graph to observe the accumulation speed of CDPs. So if I zoom on the first years, you can see in gray that the linear regression is steeper in mice than bovine and human. And to clearly see that, I spotted in 1 graph the coefficient of the equation according to the mean life expectancy. And here we can see that there is a correlation between the life expectancy and the accumulation speed of CDPs. So in other words, we can say that longer the lifespan, slower the accumulation, and inversely, shorter the lifespan, faster the accumulation. We observed exactly the same trend for CML as previously described. So to further study the carbamylation in skin, we performed an immunofluorescence study in 3 different skin sections. So a subject being 20 years old, another one 36 years old, and then an older one 80 years old. So you can see the epidermis here and the dermis for each section, and in green the homocitrullin labeling. So in the young subject we can barely see the green signal, whereas in the older subjects we can clearly see an intense signal. And this signal is very intense in the reticular dermis, not in the epidermis, not in the papillary dermis, but in the reticular dermis. And this dermis is very rich in collagen, and elastin, so two matrix proteins which have a long half-life, so around 20 years old for collagen and 70 years for elastin. So that's why we purified the type 1 collagen in skin from the three species as previously, and we observed similar results than observed with the total extracts. So meaning that there is an increase of homocitrulline in this species and a significant increase. And this increase was about 5-fold in mice, 11 in bovines, and 3-fold in humans. And as previously, we can observe also an increase of the CML, but in a less extent. So type 1 collagen is progressively carbamylated with age and accumulates in the skin. And as previously observed for the whole extract, there is a correlation between the accumulation speed of carbamylated protein and the mean life expectancy. Then we collaborate with Christian Schmelzer from Germany to perform a proteomic analysis by spectrometry on the skin type 1 collagen samples. So he did this analysis on 4 samples, and these samples were extracted from subjects being older than 77 years old. And we observed that per person and per molecule of type 1 collagen, there are 31% of total lysine residue which were carbamylated, 20% were glycated, and 14% were either carbamylated or glycated. And previous data showed that for lysine residues, so around 40% of total lysine, were enough to impair the conformation of the fibers and also to decrease the thermal stability and the fibrillogenesis of type 1 collagen. So altogether, we can speculate that carbamylation is likely involved in the skin rigidity because of the disorganization of the type 1 collagen fibers. For the elastin, we got the result just for human. Unfortunately, I don't have the results in mice and bovines due to protocol limitations. So we clearly see here again that homocitrulline increased over time in human as well as the CML. So again, carbamylation is likely involved in, or probably, this have to be addressed, involved in the elasticity loss of elastin fibers. So from this data, we were wondering if the carbamylation can be prevented by the organism. So as preliminary results, we used a mice model. So the mice were fed 3 weeks with cyanate, so supplemented water for 3 weeks, and then we replaced the water by regular water for 3, 6, or 9 additional weeks. And as a control, some mice were fed just with regular water for 12 weeks. So first, we can see that when the mice were fed with cyanate, there is an increase of the homocitrulline concentration in plasma, skin, and type 1 collagen, as expected. But then, when we replace the cyanate water by regular water, there is a decrease of homocitrulline concentration. So almost, this decrease was almost complete in plasma, and that makes sense because the plasma proteins have a short half-life, so they renewed very quickly, and that explained this huge decrease. However, in skin and type 1 collagen, this decrease is weaker, so around 70% in skin and 45% in type 1 collagen. And also this makes sense because type 1 collagen has a long half-life. But there is a decrease just within 9 weeks. So we can speculate that some system processes are existing in the organism to prevent carbamylation. So we can speculate for the intracellular CDPs that they are degradative system by proteomic system. For example, proteasomes or autophagy. For the extracellular CDPs like type 1 collagen, we can hypothesize that they are subjected to proteinases to degrade them, especially because it has been described in our lab previously in vitro that carbamylated type 1 collagen is more sensitive to gelatinases like MMP2 and MMP9. And finally, we can speculate that some repairing systems are existing in the organism. So, bottom line, carbamylation is a physiological process and a hallmark of skin aging. It's a major non-enzymatic post-translational modification as relevant as glycation. It impacts particularly matrix proteins, which have a long half-life, like collagen and elastin, is responsible for the accumulation of this carbamylated product in the skin, and they can be eliminated from the organism. Of course, these preventing systems are puzzling questions that have to be addressed in further experiments. I would like to thank my team from Reims and also the team in Germany for having collaborated with us. And I would like to thank you, and I will be glad to answer to your question. Thank you.
Thank you very much for this excellent presentation. First question, perhaps, are there a pathology, existing pathology who has to do with carbamylation?
Yeah, I didn't talk about pathology, but it's true that carbamylation was investigated first during chronic renal failure because there is a hyperuremia, so that promotes carbamylation. So that's why carbamylation was completely unknown during aging.
Thank you. Is there questions in the room?
My question is related to your answer. If there is increase in carbamylation with aging, is it because there is an accumulation of urea during aging?
So urea is stable. I mean, if we don't have any pathology, kidney pathology, I mean, the urea is stable, kind of stable. So definitely is probably because there is accumulation of long life carbamylated proteins. Because we quantified the homocysteine in plasma. For example, in mice, and there is no increase. And as I said, it's logical because the plasma proteins are are quickly renewed. So the accumulation is explained just by the carbamylation of long half-life proteins.
Question?
Thank you for this exciting presentation. The CML, the carboxymethyllysine modifications are, in the human skin at least, somehow connected to UV sun exposure. Is there something similar? Or did you check for body site for your collagen samples, or is there
Yeah.
any connection to
So
exposure?
we had the same question, but we kind of knew the answer because carbamylation is not due to any oxidation reaction. But yes, we extracted skin from the belly, from the back, the forearms, the leg, and we didn't see any difference according to these different sites of the body. And as I said, it's not related to oxidation, but to urea. So, yeah.
Marek.
Okay, thank you for your talk. So I have two questions. The first one is, so do you know if this kind of mechanism could be involved in carbamylation of neuroproteins in the skin or in any kind of tissue?
If there are other tissue that...
You focus on collagen, for example. So
yeah.
Could you imagine something that could be relevant to translate this kind of approach in other type of neuroproteins that could be involved also in peripheral tissue, like alpha-synuclein for Parkinson's disease and beta-amyloid for Alzheimer's disease?
So I don't think that any studies has been done on this kind of pathology. However, as I said to someone in here, I extracted skin from many subjects from the Forensic Institute, and I remember that one of them was a little girl. She had epilepsy, and I remember that the concentration of homocysteine was very, very high, as high as chronic kidney failure patients. So I don't know why. I just removed this data from my experiment, but it was very interesting, and I don't know the answer, but I think there is a link, I don't know, with some neuropathology. So, it was very, very interesting. So, but so far, no, carbamylation is largely investigated in chronic failure.
Okay, thank you. I see that there is, in the fields of your global collaboration, some people coming from a pharmacological institute. Which kind of pharmacological approach could you have on these fields for the future?
For the future, so different approaches can be done. So either a competition approach, so we can, for example, take amino acid to compete with our own proteins. So I think it's on the investigation right now. Another approach will be to prevent the first step of the reaction. So we can, for example, chelate the isocyanic acid and see if there is a prevention of carbamylation or not. To screen? This is, we don't know yet. So it will be interesting to study that.
Question from Marek.
Well, I don't know whether this is a question or just a comment, but it's spectacular to see that the process, which is supposed to be a chemical process, is tightly related to the lifespan of the animal or human being. It must be a kind of dynamics behind this, which is not related to the chemical interactions in the given organ, but as you say, some systemic probably changes that influence these peripheral modifications.
Yeah.
how to approach this?
Yeah, we were wondering the same question because it's just a spontaneous reaction, right? So first in mice, in the uremia in mice is a little bit higher than in human, maybe explaining that this accumulation is faster in mice. And then we can speculate that in human, because the accumulation is slower and we have a longer lifespan, so we can speculate that our organism has more anti-carbamylated system, like antioxidants that we have, or anti-glycation, system. So we can speculate that we have the same system for carbamylation. And I mean, we clearly see that for the type 1 collagen or the skin, for example. So, yeah. Mm-hmm.
Okay. Thank you very much. There is a question.